What I thought I would do is try to provide a little summary as well as what I think about where we are as a community and where progress lies. Just briefly here, my disclosures. So where are we now? As mentioned several times today, we've gone through a pandemic. We've had difficult times, but there's been terrific progress. And we have now a first gene therapy for childhood cerebral ALD approved by FDA, but no commercialization outside the US. We have a first international AMN trial completed, which is a phenomenal accomplishment in of itself, a testimony to really being a global international community. We have trials starting now 2023, gene therapy for AMN, small molecule therapy for adult cerebral ALD, symptomatic therapy for women, new model systems and targets emerging that you've heard about today, newborn screening in most states of the US and pilots in Netherlands, Japan, and Taiwan. If some of this is outdated, forgive me. I think the current trial and treatment landscape for ALD a year ago looked like this. It's in large part still true. We have certainly a known treatment for adrenal insufficiency. We have made advances in bringing ex vivo gene therapy forward. We have multiple trials that I just mentioned. We're hoping to sustain some of the early efforts in the preclinical realm that we just heard about on the earlier industry advisory panel. But a huge problem, I think, that we're still facing is a lack for treatment trials for women, and this is where we want to make a big difference. So what I'm going to do here just briefly is overall think about the landscape of disease biology and how I see the various different upstream and downstream targets in a disease to maybe remind us about the gaps we might and things we might be missing. So not to speak about what we have, but what we might want to think about. And this is actually a rather generic picture going from DNA gene defect to RNA level, reduced protein or enzymatic function, leading to lipid buildup or lipid loss, and then all the way down to organ dysfunction, whether that be due to oxidative stress or mitochondrial dysfunction or inflammation. And at every one of these steps, you can think of a different approach and a different intervention ranging from gene therapy to now the exploding world of ASOs and RNA therapeutics, down to things that have been used for longer, enzyme replacement or substrate reduction. But continuing innovation in small molecules is very exciting. So I'm just going to quickly highlight and summarize some of the things you've heard in gene therapy, which is that even with a single gene disorder such as ALD, you can have different gene therapy approaches because the biology differs in phenotypes such as childhood cerebral ALD versus AMN. For one, ex vivo lentiviral gene therapy for the childhood cerebral form of the disease, in the other, in vivo AAV gene therapy for the spinal cord form of the disease. And this has to do largely with the target cells and structures that need to be transfected by gene therapy. You've heard about FDA approval for ex vivo lentiviral gene therapy, and really the process of bringing the community together to make that happen, but also the ongoing challenges that we face as we celebrate some of the efficacy. We are also facing concerns over myelodysplastic syndrome and trying to understand how lentiviral insertion has caused dysfunction in a small minority of these boys in their bone marrow. Can AAV-mediated gene therapy help gate and balance? And you heard on the industry panel about the efforts from Swan. Just to briefly highlight, this is an intrathecal AAV gene therapy approach with preclinical proof of concept in mice, now moving forward into the clinical realm. And I'm very excited that my former mentor, Bob Brown, who's now at UMass, is together with Mehdi Ghazemi leading this trial that's about to start. So something that we will certainly learn from and will hopefully provide proof of concept that we can deliver the gene into the right compartment. Moving on to antisense oligos and RNA therapeutics. And just to highlight, I'm going into realms now that we've not talked about, just to also keep this field a little bit exciting and not just talk about things that we know about, but things that we should probably be thinking about. So new strategies to restore ABCD1 function in women is, I think, a theme that's going to start emerging and faster than we are thinking about right now. It's certainly a topic within the field of Rett syndrome and partial reactivation of the inactivated X chromosome to restore ABCD1 expression using the women's own native genes seems very attractive and something that Jenny Lee at Harvard is now thinking about for women with ALD. We know that early in development, random X inactivation occurs and has largely to do with a certain site on the X chromosome that is called exist, from which RNA spreads and inactivates the X chromosome. Can that now be reactivated in the case that you want to have a healthy dormant X now come to the rescue? Is there another approach in terms of substrate reduction or using antisense oligonucleotides beyond that? And here, just to summarize something that you've already heard about today, is that other genes may be playing a role in lipid metabolism of ALD. You heard briefly just a nice zebrafish story this morning, where an important enzyme that redirects saturated fatty acids to monounsaturated fatty acids, namely SCD1, could become a therapeutic target. And work by Josh Bonkowski and Stefan Kemp, nicely presented by Quentin Ross today, showed that a zebrafish drug screen identified compounds that improve motor behavior, and these compounds cause a significant increase in SCD1 expression. So is this a path that can be exploited, not necessarily with the drugs we know of right now, but maybe with a more targeted approach that is less toxic? And Stefan will forgive me if I remind people of his wonderful work on ELO-VL1 inhibition, and it's certainly something that's still on my mind after all these years, because the elongation of very long chain fatty acids is such a critical pathway within the pathophysiology of adrenoleukodystrophy. This biochemical hallmark of ALD, very long chain fatty acids, would not come about if there was not an enzyme that was synthesizing very long chain fatty acids, adding on those carbons. And look here at ELO-VL1 performing that critical last step. So is there a way to here target this again in an improved way beyond what has been done so far? I know that in the past there have been in vitro proof of concept with benzofibrates and others, and that have not been successful in animal models, but I sense that there is still work to be done to further improve inhibition of ELO-VL1. Just briefly about small molecules that you've already heard some about in the industry panel, but I'm just going to present some other novel targets that I'm excited about as I think about pain and mechanical sensitivity that plagues a lot of adults with ALD. And I think of all the women that come to my clinic who are all complaining of various forms of pain. And in the ALD mouse, we interestingly found upregulation of certain DRG mechanosensitive channels such as piezo2. And knowing that piezo2 has become such a focus of recent biology, in part due to the Nobel laureate last year receiving the prize for a discovery of piezo2 channels. And could blocking piezo2 lead to improvement in mechanical allodynia? At least there's a suggestion thereof in our ALD mice. Much work still to be done. Lastly, to mention some of the nice work done here at KKI on targeted brain delivery of dendrimer 4-phenylbutyrate that improves neurological deficits in ALD mice and impacts body weight. Christina Nemitz-Merth here, and also the coincidental approval of an ALS drug, just one letter difference, not ALD, but ALS, that is also a sodium phenylbutyrate and taurursodiol combination. So I think there again might be reasons to look a little across the borders of our individual diseases to see are there opportunities coming our way that might benefit our population at large. Just quickly to highlight that model systems you heard about from Joe, Quentin, and others here are really important in finding new targets for therapy. And then, of course, Omar mentioned the glio-CTN, which I think would be a wonderful way to then take some of these new compounds and feed them into a trial network that could give us answers and really advance the field for ALD. I think a huge breakthrough has been newborn screening in the US, and having now more than half of the US population being screened for it has brought huge advances. It's also brought challenges that Stefan is facing with interrogating benign variants, but it is also posing a challenge to us as ALD Connect. How do we really reach a community that is now truly national, because there are children now being identified in all states where there might not be specialists in ALD. One first step into closing the gap in care may be to look at the endocrinologist locations that here in this graph are overlaid with the 2010 census population count by US counties. Note here how for endocrinologists there is already a sparsity, with most concentration here on the east and west coast, but I think even knowing who the endocrinologists are might be a first step to providing care. Keep in mind that when you go to the pediatric endocrinologist, this becomes an even sparser pattern. But it is here a very logical partner in my mind to optimizing care for patients who are afflicted by ALD. Lastly, if we think about the global population, we have to remember that there are a lot of countries outside the US and EU who do have patients with ALD, and we've been interrogating the literature here to find population-based reports on ALD within the 30 most populous countries. And I'm struck with the fact that really most of what we focus on here has been in the US and the EU, but many of those physicians and scientists who are working in China or South America and other parts of the world might be good partners to accelerate progress with, to share insights, and to understand how care might be provided in other parts of the world. So I'm here at the end, because I don't want to keep you here too much longer, but just to summarize, there is a need for new therapies in ALD. That this need remains high. I really don't want people to leave here thinking, oh, we have a first FDA-approved therapy that is ALD is taken care of. That's not the case. It's only a beginning. Not one pill will cure all. Even within the same phenotype, there are various different stages that will respond differently to different medications. We need to really think very broadly about many different targets within this disease. Different phenotypes and disease stages require different approaches. Likely combination therapy in the future. And there's much, I think, that we can gain here. Risk-benefit assessments across all disease stages are tantamount. We've often been thinking about natural history studies, but really understanding risk-benefit at any stage of disease is what we really need. And progress remains exciting, but brings disparity that needs to be overcome. I'm struck with the fact that as we take two steps forward and make progress, we are really also leaving many others behind. And that requires really the community at large to help overcome that. So providing access to new treatments is central to our community. And I think the biggest date is tomorrow, where the Patient Learning Academy is coming to us. And so you've just heard physician scientists talk. What do they know? So it is up to you tomorrow to tell us what is missing. And I hear from Dean Sewer that all of these videos will be up shortly. And so for those who cannot get enough of this, you can go and listen to more of these talks. So I'm glad to take any questions. Thank you.
Okay, thanks, Florian. That was a really great wrap-up of today and an overview of where we're at just at large.